great research is avian chemical signals. Like all of other life on the planet, birds are capable of sending and receiving information about themselves and others using chemical signals. One of the most important sources of these signals are preen oil secreted by the Reproductive gland, which is located here above the base of the tail. This oil is used by birds while training their feathers. It helps protect the feathers from exposure to the environment, from ectoparasites, and it's also a source of volatile and semi-volatile compounds used by the birds in chemical communication. The primary study species is the dark-eyed junco, and we've been studying about 15 to 20 different volatile compounds used by these birds, and all of these compounds increase in birds who are in breeding condition compared to birds who are in winter condition, which suggests that they might be important in reproductive behavior. One of our most interesting findings so far was finding that individual odor predicts reproductive success. So as I mentioned, there's quite a few compounds. We use principal component analysis to reduce the number of variables. So this is one of our principal component scores here. And on the y-axis is the number of genetic offspring produced by an individual in a single breeding season. And as you can see here, these are males. They have a significant positive correlation between this odor and the number of offspring they produced in that one season. We also find a significant correlation for females, but it's in the opposite direction, which I found fascinating. Males and females have all of the same compounds in the species, but they differ in the relative proportion of these compounds. You can think of it like perfume, and different notes are more prominent in males or females. If we look at this principal component score itself to see what compounds are contributing to it, we see three compounds stand out. These are 2-tridecanone, 2-tetradecanone, and 2-pentadecanone. What's interesting here is that these are sexually dimorphic. We see much higher proportions in males than in females. So males with more of it have higher reproductive success, and females with less of them have more reproductive success. Now the word 2-tridecanone means a lot to me, but to you it probably gets lost in the sea of volatile compound names. So for the purposes of this talk, those three compounds that correlate with reproductive success in juncos will be referred to as junco reproductive success compounds. And I'm just going to rename these RS1, RS2, and RS3, just to make things simple for this talk. Now more recently, we've also been looking at intrasexual competition, and we found that there are correlations between some of these compounds and individual aggression in response to a same-sex territorial intruder. Now these are different between males and females. And one of these compounds is the same one we saw a moment ago, RS3. These other three compounds are completely different, are different compounds, but they all show positive correlations with different aspects of territorial aggression. Now again, I'm going to rename some compounds. When I'm talking about those three, I'm going to call them junco aggression compounds. And we'll just call them AG1, AG2, and AG3. And to make things even easier, I've tacked on the sex that shows the positive correlation with aggressive behavior. Okay, so what I want to talk about today are the mechanisms that mediate these signals produced by the or given off by the birds. An obvious target of our investigation is steroid hormones. This is a cross-section of a preen gland, or a visual gland. Go back, please. And around the edges of both the lobes, there are tubules where the preen oil itself is produced. Now, if we look at a diagram of these tubules, you'll see these little dots are actually representing androgen receptors. So this preen gland is full of androgen receptors. And as I mentioned earlier, these compounds are in much higher concentration in birds in breeding condition. So here we have males, orange is in breeding condition, gray is in winter condition, and you can see a much stronger concentration. We also know from data I'm not showing here that the changes in the early season of the breeding season tracks changes that we know happen in testosterone. So it's a likely candidate for driving variation in these compounds. So to test this directly, we did a captive experiment with birds, with male and female juncos, and we implanted testosterone, crystalline testosterone, and silastic tubing implants under the skin. We also had control birds with empty implants, and tested whether this recreated, these were birds in winter condition, and we tested whether this recreated breeding season levels of the different compounds. Well, we did see an increase in some of the compounds, but not all, and only 4 showed significant increases. And overall, this does not replicate the pattern that we see in breeding season birds. But what's interesting is which compounds are increased here. So two of the significant ones are two of our aggression compounds, AG1 and AG2. And you can also see our reproductive success compounds show no increase in response to exogenous testosterone. Now, when you're looking at endocrinology, circulating hormone levels are only part of the equation, so we also looked at the androgen receptors themselves in the preen gland. Here we did a field experiment where we presented birds with the same-sex intruder, and then we collected the preen gland and preen oil and measured androgen receptor mRNA density using quantitative PCR. And here we found only a couple of compounds in the preen oil correlated significantly with androgen receptor density. Now in males, which are here in the blue, that is actually our compound AG2. So this is the compound that we've seen a strong correlation with aggressive behavior. It also increased in response to exogenous testosterone, and we see a correlation with androgen receptor density in the preen gland. That's pretty exciting. In females, we found a different compound, and this one I don't have a name for. I haven't shown you this one before, 1-heptadecanol, because it's never shown up in any of our studies as being correlated with anything of interest before. But what's kind of cool here is that 1-heptadecanol in females correlates with androgen receptor density, and it also seems to track which stage of incubation the females were in when they were collected. All of these females were incubating, but they were in slightly different stages of incubation. So this bar graph is showing heptadecanol, but the graph that would show you androgen receptor density is identical to this. So it's a nice correlation. So it does seem to be, it does seem that androgen receptors might be mediating the relationship between a female's reproductive stage and the chemical signal she's getting out. The second mechanism I want to discuss today is symbiotic bacteria. My colleague Kevin Tice and I recently characterized the junco preen gland microbiome, and we found that junco preen glands harbor very rich and diverse microbial communities. Now we know that microbes produce a lot of, nearly all of the compounds. From the literature, we know that these compounds can be produced by bacteria. So we wanted to know whether the bacteria that we find in junco preen glands is actually producing these volatile compounds we've been measuring. So this is a list of most of these compounds I've been talking about. And what we found, two of the most common bacteria that we find in junco preen glands, this is in over 3/4 of all of our samples, are Burkholderia and Pseudomonas. And these two genera are known from the literature to produce over half of the volatile compounds that we measure in juncos. Now just because you see a dashed line here doesn't mean that it's not produced by any bacteria, it just means it's not produced by those two genera. And I just want to focus in on our two sets of compounds I've been talking about. The 3 reproductive success compounds, it does appear that it's possible that at least 2 of them can be produced by these two genera. On the other hand, if we look at our aggression compounds, there's very little evidence that they would be produced by these bacteria anyway. And one of them, AG2, the same one that was showing all the interesting correlations earlier, is the only one on this list that's not known from the literature, from the database that we were using, to be produced by any bacteria at all. So there's not much support that that set of compounds were produced by bacteria. But now we want to test whether these 3 here are in fact produced by bacteria. So we conducted an experiment again with captive juncos, and we injected a broad-spectrum antibiotic and rofloxacin directly into the preen gland of half of our juncos and saline into the other half for 5 days, and then measured the volatile compounds in the birds in the 2 treatment groups. And we found only 3 of the compounds were reduced by antibiotic treatment. And what's really cool here is that these are our 3 reproductive success compounds. So you can see that birds that received antibiotics had significantly lower concentrations of these 3 compounds than birds who were in the control group. There was no significant effect on the aggression compounds. So to review, we looked at 2 different possible mechanisms for producing these volatile compounds that we think are important in reproductive behavior. Physiological mechanism, specifically looking at androgens and androgen receptors, and the microbial mechanism, looking at bacteria in the preen gland. We find evidence that one of our aggression compounds is correlated with aggressive response to a same-sex intruder, there is evidence that these might be related to both circulating testosterone and androgen receptor densities in the cream gland, but no evidence that they might be produced by symbiotic microbes. On the other hand, our 3 reproductive success compounds, which are correlated with both male and female reproductive success, are produced by bacteria, we know this from the literature, and are significantly reduced by antibiotic bacteria in a captive experiment. So I find this to be an incredibly exciting finding, that we've got a potentially sexually selected signal that's produced not by the animal itself, but by its symbiotic bacteria. And so we've got a lot of exciting new questions here that we want to address. So first of all, we do see that physiology and microbes can both influence juncochemical signals, but next we want to know how these factors interact. Does an individual's hormone levels affect what bacteria it contains? Does a genotype affect what bacteria they're harboring, and so on. Particularly interesting to me right now is what are the relative roles of genotype and environment in shaping individual chemical signals. We know from data that we're writing up right now that birds share their bacteria through physical contact, especially from mother to offspring and between mate pairs. How does that affect their chemical signals? And what are the implications of all of this for understanding sexual selection on these signals? So I'd love to hear ideas from anyone throughout the rest of the conference. I've got my email here and my Twitter handle. So now you know why that's my Twitter handle. And I'd like to thank my co-authors, the many, many people who helped us out in the field and in the laboratory, and my funding sources.